Today we are going to discuss another topic on uh, continuity of life, and to discuss this topic we are using uh, example from somatic cell, human any cell, and to compare the division process we have started specifying male and female with their genetic setup. So here you can get 46 chromosome, 22 autosomal pair and X and Y for the male, and 22 autosomal pair XX for female. So as you know, you can get this sort of germinal cells in the testis in case of male, and in seminiferous tubule spermatogonia, and in the female there is a specific organ, ovary, that contains egonium cell. So to make the student understand the concept, we have used somatic cell and their diploid chromosome, that if any part of the cell DNA is under stress or damaged by any rays or any mutagenic effect, this cell will vigorously divide. So now the question arises, what the division is called and how does it related to us? So division means when a particular cell will divide. So before it goes, it perform the division, it has to follow a certain standard pathway which is interphase, that is G1, S, G2 and M. And M again is divided into prophase, metaphase, anaphase, telophase. And after that, one cell will divide into two new cells. But the question is that if when your cell has only 46 chromosomes, how will they divide into new 23 pair containing or chromosome containing 46 chromosome containing cell? So to accomplish this task, they have a special machinery called, or a special phase in their interface is called synthesis phase. So synthesis phase actually doubles the pre-existed DNA. So pre-existed DNA, in case of the human being, we call it BDNA, and BDNA is a right-handed helix. Like this, you can easily understand it's moved like this. So right-handed helix and 10.5 base pairs in a complete turn, and the total length of it 34 angstrom. You will divide simply, you divide, it is 3.4 angstrom. The pitch rise, the subsequent nucleotide pair. One of my students have asked me, what is a nucleobase pair? So to make them understand, I have used AT pairing. So you can see here adenine hydrogen bonded with hydrogen. So here you can get a base pair. So we used the example of human genome. There is more than 60,000 genes within this whole 46 chromosome. But for our understanding how it divides, we have taken a small part of BDNA. But this is not the end of the DNA, because if we go into the animal kingdom or the organism kingdom, we find that viruses is also there, and they have some simple organization, morphology, and all are in 100 to 200 nanometer in length and 100 in weight. But the important thing in our science how we are going to use this bacteriophages, because bacteriophages T4 and T7 is in the modern industry, biotechnology and all, they are using T4 or T7 because it is a lighting property. And another lambda phase, so these are both double stranded linear DNA but very small, 5X174 M13. These are lysogenic viruses. So what is lysogeny and what is lytic? In short, in a nutshell, I can say that lytic means it's the landing of the phage virus, bacteriophage, and release of the blue one, release of the double stranded DNA, and then it multiplies and it ruptures the cell and move out. Now, but in lysogeny, they use to integrate, co-integrate within the DNA of the host. So in our case, we have used the host, bacteria, and our example is Escherichia coli. So how they feel suitable? Why? The bacteriophage feels that I can flourish within this bacteria. So to understand this, they have the bacteriophage in the S phase also undergo multiplication of the DNA strand. So to multiply, the E. coli use the same machinery. They have first RNA primase to synthesis this segment, we call it RNA primer. So from RNA primer, our strand is continuously synthesis, that is called leading strand. Another strand with small primer synthesis, they along the strand 5 to into 3 prime from away from the fork. So this consecutive short 200 to 400 nucleotide sequences are called okazaki fragment. So to this, polymerization is done by DNA polymerase 1, 2 and 3. So in our case, we have shown this. These are the primer, again multiple primer, but when the fork replicates towards this Y-shaped fork is called, they use helix first separating enzyme, DNA helicase. So until they use this helicase, they cannot break this bond. This special enzyme has the capacity to break the bonds. So in this way, you can find a very clear-cut theta-shaped or open space. And now, why they do not re-anneal? পুনরায় কেন আবার reunite? So reunite is not possible because helix destabilization protein, single stranded binding protein, bind to refrain for reannealing. So this part using the same machinery, using both the T4 and bacteriophage. So whenever the bacteriophage finds an opportunity to get integrated, it's flourish. Now T-phage, we have shown you, it is 169 base pair and kilo base pair. Their DNA structure is, and they are bounded by capsid protein, capsulium, as you know. And our DNA replication completes the doubles the total DNA. And this replication is completely done in our S phase. So after S phase, they will perform uh, RNA synthesis, because this RNA synthesis will form some protein and the continuity of this thing will go on. So again, the bacterial chromosome here is a circular double-stranded one. Circular double-stranded is devoid of this histone protein as we find in our eukaryotic cases. So if as there is no histone protein, it remains in a unquiet condition because it is found in our prokaryotic system in cytoplasm. And eukaryotic system, the whole thing is found within this nucleus. And DNA replication is within this nucleus. The DNA replication exists. So, and again, our eukaryotic human or any organism is VDNA, ZDNA, and VDNA, ADNA, and ZDNA. So we will discuss this thing later. And another thing, this chromatin of any human chromosome during in a or after the M phase, when you see a cell in a very resting that means without taking part, or it is in the primary stage of starting the process mitosis. Here actually this methylation of these nucleotides make it condense. So methylation means the chromatin will form now chromosome with the help of histone and other proteins. But in case of DNA replication, you need to decondense this enzyme. So for the decondensation, you require histone acetylase. So histone acetylase means a group called CH3CO need to be fixed with every nucleotide. So if you incorporate this with the help of enzyme, the condensed form of the DNA or chromosome will become loose. So it is a active state and chromosome will follow the subsequent stage of this prophase becoming one of the stages. Thank you.